Hi, I'm Callie, a postgraduate researcher here at the University of Bristol. I'm studying a research master's in biological sciences. In this short video, I will be talking through plant transformation and my research into this to help demonstrate how small changes can help us solve some really big problems. But why plants? Well, what do you think plants do for us? I'm sure you could give me some ideas. Well, here's some ideas. There's so many things that plants do for us, from providing the food that we eat to creating clothes that we wear. There are many things that probably aren't on this list that you may have thought of, but this is just to show that there are many reasons why plants are great. But what if we could make them even better at what they do? Actually, we've already been trying this for hundreds of years through traditional breeding. Traditional breeding picks the best plants that are naturally the most beneficial to us, such as those that have tasty or many fruit. For example, corn used to be tiny, with a handful of small hard kernels, but over these hundreds of years, we get the corn that we see today. Now we have a much bigger corn, a corn on the cob, which is sweet, tasty, and a bright yellow colour. Unfortunately, traditional breeding is a very slow and random process, and is unable to keep up with our rapidly growing population. It's a bit like a deck of cards. Let's say we have 4 cards that we really want, or 4 genes, and then 4 genes that we really don't want. And they're all mixed in with a whole lot of other genes, and we shuffle those together when we cross them. After mixing all these genes together, we then get a sample of them out in the progeny. So for example, we might get a whole lot of genes we don't really care about. We get 1 bad gene, 1 good gene, maybe a couple more bad genes, and then a bunch of other genes. So overall, you can see it's not exactly what we hoped for, and it's just like this with traditional breeding. And this is where plant biotechnology comes in. For this, we're going to zoom into the plant right up to the DNA. Plant biotechnology is the modern day of plant breeding, the process of directly altering plant DNA. These changes could be adding new DNA, removing DNA, or making changes in the DNA present. These tiny changes can make a real difference in a plant. We do this a lot in research to find out the effect of particular genes on a plant, but also there are a lot of real-world applications out there. Before we jump into the applications, I'd like to explain how exactly plant biotechnology works. The first main method we use uses a bacteria that naturally infects and alters plants' DNA to insert its own. So we provide the bacteria with the DNA that we want it to add. Unfortunately though, the bacteria only infects a limited number of plants, so there are many plants that this method does not work with. The second main method uses a gene gun. This fires DNA carried on tiny particles at really high speeds, which breaks through the plant cell wall. This allows the DNA to be deposited within the cell. But as you can imagine, this can be quite damaging to the plant and therefore is quite inefficient, as many cells die in the process. So although we have these 2 main methods, there are actually a lot of other methods out there. For example, a lot of researchers are currently looking into the use of nanotechnology, using nanoparticles as a new way of delivering DNA into plant cells. And this is relevant to my research today. My research looks into developing one of many new methods. This uses a nanotechnology method created here at the University of Bristol. It uses carbon nanodots, a nanoparticle, to transfer DNA into a plant cell. Carbon nanodots were chosen because of their ability to pass into plant cell walls without the need for force, and can be modified to carry DNA into the cell. Here's a diagram to give you an idea. The blue dot in the middle is the carbon dot, and the green chains around the edges are a modification that allow DNA to bind. The method is as easy as mixing these nanodots with the DNA and spraying this dilute mixture onto plants. Pretty simple, really. Now that you know a bit more about the methods, we can talk about the applications. We could use these tiny DNA changes to help plants do what they do better. Remember the many things that plants do for us in the world? well, there are endless applications for this technology. For example, we can look at the issue of climate change. We can make plants that are able to combat climate change by making them more efficient, perhaps enhancing photosynthesis. This way, plants require less farmland, and this reduces deforestation needs and reduces the impacts deforestation has on the world. We could also make plants that are able to survive climate change, for example, plants that are more drought tolerant. We could also look at food security and our growing population, perhaps making crops that produce more or better crops. But we could also look at this in a more interesting level. For example, we could look at changing the nutrition within plants. Golden rice has increased levels of vitamin A to help combat vitamin A deficiencies in developing countries, which causes blindness. There is also the possibility of removing allergens from crops, such as making gluten-free wheat. We can also look into the topic of medicine. We can make medicine production more sustainable. Instead of using factories, we could make plants that produce important molecules like medicines, using plants as little biofactories. They could even produce vaccines, potentially making edible vaccines. So as you can see, there really is great potential for plant biotechnology. So I wonder, what ideas do you have for the future of plant biotechnology? Is there a particular issue you think we could tackle with plants? I hope you enjoyed learning about plant biotechnology and that you can take away something new from this. This video was recorded for the Bristol Doctoral College's Research Without Borders event 2021. If you have the time, please go and check out some other stalls to find out more about postgraduate research going on at the University of Bristol. Thanks for listening.